Today I will talk about hypothalamic regulation of food intake. Okay, we have two cats there. One cat which is lesioned with the ventromedial nucleus of hypothalamus, wherein it produces hyperphagia, eating and eating only, not satisfied with whatever has been given to, and that is what that becomes obese and increased body weight. The other one has been lesioned with the lateral hypothalamus, which does not want to eat, and even you can see the size of the animal. It is just almost practically starving. Maybe it may die if not this thing. So that is what previously they thought. Move on. I just completed this series, the endocrine regulation, in my first lecture, where I talked about the parts of hypothalamus and connections of the hypothalamus. In the second lecture, I mentioned about the thermoregulation. I completed that. Today, I will be talking about the physiology of feeding and satiety. The rest of the other things, I will take it further. Okay, so now, these are some of the experiments done in rats before we want to understand why food regulation. So these experiments are done in a group of rats. These group of rats, the orange group, so they started, all of them started somewhere here only with this weight. And at this point, the animals were given a particular, or they were subjected to particular food regime. The orange one, or the A group, they were force-fed, and this black line indicating these animals were subjected to the normal food, and these animals were having a restricted food. Okay, this is the period of their whatever the experimental design, from around day 30 to day 50, just somewhere around 18 to 20 days of these things for the rats. When the normal, this normal one, B group, received a normal diet, they had enough diet, so there was no restriction. And these A group were force fed. They were given extra, as happening to you right now, because you are there in the house, maybe munching all through, and must be when you come here, you may have to do a lot of exercise to get rid of that fat. Okay, anyway, so now come back. They were force fed, and they increased the weight. And after some time, because at this point, number 2, they all rats were given a normal food ad lib. Ad lib means as sufficient as required. It is there, and they have to go and eat. So now, after some days, it came back to normal. You just see around 70 days, they became almost the same level as the normal, because genetically they are of the same group here. So they are here. Then second group, which was a starved or a restricted diet, they reduced the weight. So they did not increase the weight, they reduced the weight, and then again they also recovered. So that means there is what is called a phenomena of regulation of the body weight. So you just see regulation of the body weight when you are subjected to two different conditions, either excessive eating or decreased or restricted eating, these things. So that means there is some regulatory function which is responsible to keep this whatever body weight in the normal range. That is what it is. This graph tells about. So now let us consider about feeding behavior. It is similar to the temperature regulation, because it has a control system which has a increased and a decreased, that means orexigenic and anorexic activity. And however, the feeding regulation is less complete than the thermoregulation. Thermoregulation is very well it's clear and it is understandable. We can logically put them back. Now, third point, the body weight. Our body weight is the function of food intake, that you all agree. And this function of the food intake is regulated by a set point. every person is having a set point. Even if you are giving a food, he will reach up to a point, and after that it will try to decrease. So that means there is what appears to be like a set point. So then apparent set point of an individual can vary with this set point can vary with the different aspects, different parameters like stress. When somebody is stressed, maybe you are stressed, or when you have examination, you are under stress, either you may increase the eating habits or you may decrease the eating habits. So that stress is altering. Then the food palatability, as you people always think that hostel food is not good, it's never a homely food. In that case, you are always trying to lose a weight, your set point is lowered. When you are at home, maybe same food is served there, but still you enjoy that the particular surroundings. Then exercise, so that also would alter your feeding behavior. The mood, so mood in the sense, if you are having a good mood, you will eat. If you don't, then you will try to restrict, and you say that no, I don't want to eat. I don't want to eat. I have no mood. So that's exact these things, and many other environmental and genetic factors. They influence the feeding behavior. One important thing, even a small increase or decrease in daily calorie intake would alter the weight gain in a long range. You can just sit back and calculate. Suppose you are gaining 1 gram weight every day. You can imagine you have gained nearly 500 gram weight in 1 year. So I am talking very less. 1 gram is too less. Our body variations in the morning to evening, there may be 1 to 2 kilo variation is there. So that means a small increase of daily weight would increase your substantial weight for a long duration. That's what I want to impress upon you. So there should be, hence the body must be governed by signals which control the food intake and energy balance as a long-term strategy. That is about the feeding behavior. Now, let us come back to the historical experiments. Those are the rat cats which were done by Hetherington and Ransom in 1942, or similar group of workers. And they have shown that food intake is thought to be under the control of 2 regions of the hypothalamus, the ventromedial nucleus and the lateral nucleus. They built up these 2 regions because when they destroyed the ventromedial, the animals were eating hyperphagia and produced obesity, and the animals were lesioned with the lateral hypothalamus. They were having aphasia, and they lost weight, and the animal died unless they were fed. Similarly, opposite of this thing, electrical stimulation of the ventromedial nucleus suppressed the feeding, because the ventromedial nucleus is one which satisfies, or which gives the satisfactory thing, that is satiety. That is a satiety. When I stimulate the satiety center, it says that okay, I'm fine. I'm satisfied. I'm full. So something like that. Electrical stimulation of the ventromedial nucleus suppressed the feeding. Electrical stimulation of the lateral hypothalamus elicited the feeding. With these experiments, the Hetherington and Ransom, they mentioned ventromedial region and lateral region. It was later in 1950s were substantiated by B.K. Anand and Brebeck. B.K. Anand was an Indian who was a director, or who was the head department of physiology in AIIMS, Delhi. He's no more now. So now Anand and Brebeck, they proved further in 1951. So these were the first authors who mentioned about these ventromedial and lateral regions. Now, with those experiments, they postulated a dual center theory. There are 2 centers. Maybe in most of your textbooks, this may be existing. One, the lateral hypothalamus, lateral nucleus, or lateral area of the hypothalamus, and this hypothalamus is known as a hunger center or a feeding center. And another area of the hypothalamus, which is medial. This is the ventromedial hypothalamus. This is known as a satiety center. So each inhibits, when we are hungry, when we are eating, we suppress the satiety center. When we are full, when we are satisfied, it suppresses the feeding center. That is how the feeding, the short-term regulation of the feeding is governed. Because when we are full, we say that okay, enough. I don't have much. I don't want to eat much. My stomach is full. That is the type of feeling. Or you are satisfied, and whatever your hunger has been quenched. So in that way, so this dual center works. What happened? Because of certain experiments or certain things, this dual center theory did not support the following things. One, brain, especially for the food control, is not organized in discrete centers that control the feeding and the satiety. Then neural circuits regulating the homeostatic functions of the feeding and satiety are distributed in a number of areas in the brain, because it's not only restricted to the ventromedial or lateral nucleus. Then there are other components, sensory components associated with the feeding, just like a hedonic component. Hedonic component is a pleasure. Hedonic is a pleasure, which gives a pleasure. When we eat food, we have a pleasure. So that means, especially if you are eating a nice ice cream or a nice chocolate, you have the pleasure of eating that, and you try to enjoy, and you want to eat more and more of that. That is a hedonic component. That is a pleasurable aspect. So that pleasurable aspect, that sensory aspect, and another social component. Suppose you are in friends, you have gone to a party, and then everybody says, let us have ice cream. Then you also go for ice cream, though you are already the hypothalamic component, the satiety center has said, okay, well, I'm done. But still then you try to eat that extra whatever the ice cream which has been served to you, extra dessert, your social friends are forcing you to do. So these things did not explain the dual center theory. So now, in animals, upon lesions with the lateral hypothalamus, lowered the set point to maintain the body weight. Just like here, these are the non-lesioned animals, and these are lesioned animals, and these are lesioned elements pre-start. When that means lesioned where? They are lesioned in the lateral hypothalamus. Then when they, after lesioning, they were given the normal diet, so they came back and maintained a particular weight. So that means they did not go below particular weight. So that means they were falling the weight, but the weight came back to a certain level. So that means there is a bouncing of the weight, and the lateral hypothalamus alone, because we were thinking that lateral hypothalamic lesions would kill the animal. No, it did not kill. A body weight is maintained at a lower level. So that means there is something which is governing the food regulation. Now, what happened then? Subsequently, we have another other important things which do not accept the dual center theory, wherein the loss of mesolimbic or tegmental dopaminergic neurons, they will also produce aphasia. Even the lesions of the nigrostriatal path. Nigrostriatal path is for the basal ganglia, so that is from the for Parkinsonism. That also produced aphasia. That means in a Parkinsonian patient, he will be having a less. They may not like to eat. That means they try to say that no, I am not hungry, or they don't want to eat. So that means these did not explain the dual center theory alone. That means lateral hypothalamus or medial hypothalamus. Even the enteric system, that is the gastrointestinal system, the hormones from the intestinal system like insulin, leptin, CCK, ghrelin, and peptide YY or pancreatic peptide. So these are all involved in the homeostasis. They are also did not. So you can have the leptins, they would suppress the feeding. The CCK, what it does, it shows that it is full, Once the full is there, so that will suppress the feeding. Insulin, because of the glucose homeostasis, suppress the feeding. We have a ghrelin, which is secreted from the hungry stomach. I say hungry stomach. So this hungry stomach releases the ghrelin. The ghrelin will increase the feeding behavior. So that means these three or four, these three are, they will try to suppress. This will try to increase. So that is how the energy, short-term energy balance is taking place. Now, besides these these objections, thermoregulatory mechanisms, because whenever we are accepted to the heat stress or a fever, what happens? That would alter this control center. In addition, this anticipatory response is the conditioning, because suppose you have eaten your food, or you have taken something in the cafeteria, some cake or something like that, you are full, but around 1 o'clock or whatever your daily set time for the food or lunch, you will feel hungry. So that is a conditioning time is not altered. And even after exercise, maybe you have a bout of exercise and these things, suddenly you feel hungry, so you want to go and have something which is energy rich. So now other external influences, that means your friends, the circles, and the stress, so many factors, so they will also influence the either hedonic or the social influences, they will influence. Then in chronic illnesses like terminal cancers or infectious like tuberculosis, they produced a sort of a loss of anorexia, and this anorexia associated with sarcopenia, that is called the cachexia. The cachexia is the loss of weight of these persons, these patients. This is also not explained. So this is what I have just. So we have a feedback loop. This feedback loop is outside the range of the central nervous system there, and this feedback loop is within the range of the central nervous system. So now with this dual central theory is not accepted. Now again, there are so many other experiments done here. The paraventricular nucleus, up till now I did not talk about the paraventricular nucleus. When the paraventricular nucleus is lesioned or cut, and in this paraventricular lesions there are two type of neurons. One mcr4 neurons. These are the neurons who are expressed with the melanocortin 4 receptors. So when you eliminated them, there is the animals were hyperphagic, the excessive eating and obesity. And they also have these paraventricular neurons have the TRH containing neurons. These TRH containing neurons, when they are ablated or lesioned, so there would be aphasia. That means there would be again two things, dual things happening in the paraventricular nucleus also because of these receptors. Then again, we have another center there, dorsomedial, because we were talking about the ventromedial nucleus. Now dorsomedial nucleus that produced hyperphagia and obesity, the lesions of that. Then came the master here, the Arcute nucleus, where all the GIT hormones, all the hormones and all the things are converging here in this point, and that would regulate the food intake and energy. Thus, several hypothalamic nuclei participate in food intake, and they are: one, ventromedial nucleus, lateral nucleus, arcute nucleus, paraventricular nucleus, and dorsomedial nucleus. Now, if we consider these, so now I have put them on this whiteboard there. So that means we have a paraventricular nucleus here. We have a arcute nucleus. This is the arcute nucleus. This is the arcute nucleus, and we have a ventromedian nucleus, and this is the lateral hypothalamus or lateral nucleus. So these are the old classic one. Now what is coming? The circle is the main thing which is steering. That's why I've used that steering wheel. So this is steering all our feeding and satiety. So now we have ventromedian nucleus and dorsomedian nucleus there, and these are the things. And we have the sensory modalities regarding the food, the palatability, the smell, the taste, all components, the settings, surroundings, and all those things regarding the food, the conditioning component, and we have the um, prefrontal cortex. So that also plays, that is a social prefrontal cortex. So then emotional amygdala, and this is a reward center nucleus accumbens, and the hippocampus is the memory center, which is in association with the amygdala. They will all govern the food intake. Now, ghrelin is a hormone which increases the food intake. Leptin, insulin, CCK, peptide YY, and pancreatic polypeptide. So they all suppress the food intake. Now we have a Rafi nucleus, that is serotonergic, wherein vomiting center is located. So that would suppress the activity. The vagus tells about, because when the stomach is full, vagus tells about the state of the gastric distension. So that means that will suppress the food. The NTS is another thing which will talk about eating of the food or stop eating of the food. Then parabrachial nucleus, locus coeruleus, the anterior pituitary. These are things which all these units, they are working for the feeding and satiety. Okay, so now we move on to this thing. Now, what we have, because in this entire thing, I have made this steering wheel, and this in this steering wheel, that is the arcute nucleus. This arcute nucleus has a two set of neurons. One is a neuron which is expressed with a agouti related peptide or protein and neuropeptide Y. These are excitatory to the feeding, so they will increase the food intake. That means orexigenic. So then we have a POMC, pre-opiomelanocortin, and CART. These neurons, they are inhibitory. So each of these neurons are inhibiting one another. So this AGRP neurons inhibit POMC, POMC inhibit the AGRP neurons. So that is why I have made a bidirectional these things. So this is having a GABAergic input there. So now that is what is coming up here in this graph regarding this AGRP neuron. AGRP neuron projecting it to the neuron. This is the arcute nucleus. This one is the arcute nucleus, and this is the paraventricular nucleus. And from this paraventricular nucleus, here is the lateral hypothalamus. Okay, so now what happens here? The AGRP neurons, they are activated by ghrelin. Ghrelin gives it, because you have the ghrelin receptor there. The ghrelin activates it, and once it activates the neurons in the paraventricular nucleus, and these neurons in the paraventricular nucleus, that would activate the food intake. Okay, so now at the same time, these are having a inhibitory interneuron through the GABAergic interneuron. They will be inhibitory to the POMC called neurons. So this cocaine and amphetamine regulated transcript. Cocaine and amphetamine regulated transcript. These neurons, they will inhibit. And if you're looking, this is a plus, this is a minus. This will decrease the food intake. So this whole thing is balanced within the arcute nucleus. One. And this is also balanced here in the paraventricular nucleus. That is what I was talking about. When these things are activated, they will increase. That means if I am going in this path, increase the food intake, and it will activate the nucleus NTS. And this NTS activation increases the sympathetic activity. Sympathetic activity is energy loss or energy expenditure is happening. So that is how the food is taken, energy is spent, so balance is maintained. Now we have another things here: insulin, leptin, and this thing. Now let me come back with an example of the diabetes mellitus. What happens in diabetes mellitus where there is a lack of insulin? And insulin is required for the cellular glucose level within the cell. Now, when the insulin is not there, the glucose is not reaching. So that means if there is a decreased insulin, this will inhibit the AGRP neuron, and it will excite the POMC CART neuron. Or if there is a lot of circulating leptins, because the leptins are coming from the adipose tissue, Or if there is a CCK, that means the CCK is released when the stomach is full and the fatty diet is being digested in the first part of the released into the first part of the duodenum. So in that case, that would suppress this eating, and that would activate the non-eating, that is a anorexic activity created by the POMC or CART. This will be inhibitory to it, and this would inhibit this, so that once it is inhibited, less food intake and less body expenditure. So that is what I have mentioned here. You can just read these things and whatever I have explained. Now we are moving further. Now, if the whole thing, the AGRP neuron, if you are looking at that, this AGRP neuron is very complex, very complex. Now I am just making it simplified in the sense I don't want to go further, because I could have taken only on AGRP neuron today. But anyway, I'm not interested because it's too beyond your these things. But anyway, I want to mention that AGRP neurons are connected with the paraventricular hypothalamus. They are connected with the lateral hypothalamus. So that means there is a set, these neurons have a bilateral link with this. Then they are connected with the stria terminalis. That means alpha, that means anterior bed nucleus of the stria terminalis. Then they are connected with the parabrachial nucleus in the reticular system. They are connected with the central amygdalar nucleus, that is a behavior. They are connected with the periaqueductal gray. They are connected with the thalamus, the paraventricular thalamus. So that means they are, because this part is connected further to the cingulate gyrus or the prefrontal cortex or other hippocampal areas. So that is how the entire thing is AGRP neurons are connected. I will not go further. So this will be about the AGRP neuron. And now I come back with whatever those things I had made there. Now I want to put them here in one by one. Let us start with the ghrelin. When I am hungry, so that means when there is no food is taken after a period of some amount of time, the stomach is hungry and hunger contractions began. The hunger contractions began. So these hunger contractions release a peptide hormone ghrelin, and this ghrelin reaches the hypothalamus, especially the arcute nucleus. And this ghrelin is coming as a hormone, and at this part of this thing, the blood-brain barrier is broken at a particular areas in the brain. The blood-brain barrier is not intact. They will reach to the site, or even with the blood-brain barrier, ghrelin crosses the blood-brain barrier. So now, once the ghrelin reaches the AGRP, now this AGRP are neuropeptide neurons that will activate them. The activation of ghrelin activates the lateral hypothalamus. Now lateral hypothalamus indicates the hunger, and you will search for the food. Suppose if there is no food, it will tell the dorsal motor nucleus no food. So now it will talk to ventromedial nucleus, and it says that, come on, you are hungry, but the food will be coming after half an hour. So that will suppress. This is what I am talking about immediately. Now let us not bother. So this is one aspect of the ghrelin. Ghrelin has started the game. So that means you go for the feeding. This is hunger. I mean to say that feeding. So then you have this ghrelin the AGRP neurons are connected with the paraventricular neurons. Now they will activate a TRH containing neurons in the paraventricular nucleus, and these TRH containing neurons will activate lateral hypothalamus. They continue to eat. They continue to eat further. Okay, that is the second aspect. So now what happens when it's going, it gives a collateral to the dorsal motor nucleus again, dorsal medial nucleus, sorry, dorsal medial nucleus, and these are NPY containing neuropeptide Y containing neurons. They will activate again another bout here to the paraventricular nucleus, and that would again make this thing. If you are looking at the yellow, the blue line, this blue line is for all feeding. So this would again activate it will activate the lateral hypothalamus. That is how you continue to eat. So that means you started eating there. This is how it is began. Then after started eating, this is happening. Meanwhile, what is happening? The eating is not only just a food. It is not bland. So there are senses associated with the food. There are conditioned reflexes associated with food. There are social aspects, the prefrontal cortex associated with the eating. The emotional aspects associated with it. Suppose your mother is serving you a food, you eat an extra serving. And the reward aspect, because you think that maybe you will be rewarded for something, or all these things also add to the lateral hypothalamus, and they will also stimulate the AGRP neurons. So these are the whole thing. They are participating in the orexigenic or orexinergic neurons. These are all enhancing the food intake. Now what happened? So I have started eating food. I have eaten food, and once I have eaten food, so that means already the stomach is full. Once the stomach is full, that fullness of the stomach, because I've started eating food because the blood levels of the glucose has gone down, the insulin levels are higher. So now once the blood glucose levels are being this thing, so that means so many things are happening. Let me start one by one here. So now what happens? Insulin levels increase, because after eating the food, insulin levels increase. Once the insulin levels increase, this would suppress, this would inhibit the AGRP neurons, and this would activate the POMC neuron, the insulin. So now, if it activates this thing, it will activate the ventromedial nucleus. I'm taking the ventromedial nucleus. Now, ventromedial nucleus in turn suppress the feeding. So this is how it is happening. So that means started here. So it suppresses this. Once it suppresses this is not inhibited. This is activated. This would go to ventromedial nucleus, and this would be the inhibition. Now, second point, what is coming back from the ventromedial nucleus? It is suppressed. So that means the AGRP neurons are suppressed again. Further, the AGRP neurons, if they are activating the insulin, activates this POMC neuron that I have not shown there, because to make it clear. So POMC are called neurons, and these would activate the MCR4 receptors. And these MCR4 receptors work for this red line there, that will activate the uh, dorsomedial nucleus. Now, this red line will activate the ventromedial nucleus. So see another red line here. Now, this red line that suppresses the feeding. So that means that is how the feeding is just stopped. You say that, well, I am full. I am full. Okay, now what happens? You are full there. So this is one aspect about insulin. Similarly, leptin does. Similarly, what happens when the food enters into the first part of the duodenum? CCK and PYY are released, and they are again doing the same thing as the insulin did through the anorexic path. Anorexic path is the red path. So they will activate. This is one aspect. The second aspect, what happens here? So the stomach is full. The vagus is activated. Vagus is activated through, maybe it's coming through NTS or vagus coming to the medulla. That would suppress because the stomach is full, you don't want to. If it is too full, it will say that, come on, stop it. Suppose if it is too full, then rough nucleus is activated. What it does, it will try to throw it out. So that means it stops here, then there is a vomiting. So if you keep on eating and eating and eating, and then you will throw out. So that means that is one of the aspect. Then parabrachial nucleus is concerned with the hedonic component. So that will be concerned with the amygdala and hippocampus and nucleus accumbens. So they will also try to restrain. They will be inhibited. They will try to restrain the activity. Similarly, the reticular system, locus ceruleus, the norepinephrergic system, they are inhibited. Similarly, the hormones released from anterior pituitary are also really decreased. They are all participating in the anorexic pathway. So you can just see that, especially the hormones are the corticotropin, what we call it as ACTH. So that would suppress or MSH. They will suppress these neurons. This is a melanocyte concentrating hormone. That means alpha MSH will activate this and that would produce. Okay, so this is all the story. Now I want to just make so whole center is situated in the steering wheel of the arcute nucleus, and this arcute nucleus divides or decides whether it is going to the feeding or hunger, or whether it is going to the satiety. And here itself there are two set of neurons. They will have a local regulation. Then second, this one is dependent or the assistant for this thing, or the second one who assists is the paraventricular nucleus. Again, there are two set of neurons, one TRH expressed neurons and MCR4 expressed neurons, which are inhibitory to each other. So they will again drive you for orexigenic and a non-orexigenic path. So now dorsomedian and ventromedial nucleus, they are sharing certain responsibilities together, and they will suppress the lateral hypothalamus. This is all the story of the eating, what we do, and the regulation of the eating. Now let us move further. So this one I have in this graphs, the same graph I have mentioned, but I have given the details of these abbreviations just for your benefit, because I have explained all those things here in this and the abbreviations, because I cannot put all the abbreviations here. I have made it a point to give you all the abbreviations because you may be wondering what they are all about, and you can read and you can get it through this. Okay, we move on further. These are the tables showing the various transmitters or hormones which decrease feeding or increase feeding. The ones which decrease feeding are known as anorexic. The ones which increase feeding are orexigenic. So now anorexic are the alpha melanocyte hormone, leptins, serotonin, norepinephrine, corticotropin releasing hormone, insulin, cholecystokinin, glucagon-like peptide, cocaine and amphetamine regulated transcript of the SCART. So that means amphetamines, they decrease. So amphetamines were tried sometimes the drugs to reduce the weight. So then peptide YY and neuropeptide, these are all anorexic. So now orexigenic, that means which increase the feeding, or neuropeptide Y, agouti related protein or peptide, melanin concentrating hormone MCH, orexins A and B, endorphins, galanins. Endorphins are the opiates, and galanin and glutamate and GABA, cortisol, ghrelin. So these all increase the feeding in the hypothalamus. Now again, I make a very simplified summary of the feeding and satiety, giving a simplified diagram. I don't want to clutter you with all the things. Here I have the arcute nucleus. This is the arcute nucleus here, and in this arcute nucleus we have the AGRP and POI neuron, and we have the POMC CART neuron, and this will activate the lateral hypothalamus. Lateral hypothalamus brings about the feeding or hunger, and through the paraventricular nucleus and lateral ventricle they bring the hunger. Similarly, the activation of these, because whenever some of these neurohumoral factors which I have mentioned who activate them, especially leptins, insulin, CCK, when they activate, they will activate here, they will indicate about satiety. And besides these, the various brainstem, the cortical, amygdalar, prefrontal, and various other sensory inputs also influence these NPY or POMC neurons to bring about the hunger and satiety. This is summarized. I don't know nowadays the concept of just ventromedial nucleus and lateral nucleus is gone. It is the era of the arcute nucleus which is taking up. Okay, now I pause here. I think I now consider the various disorders of feeding and satiety, especially the disorders associated with the feeding and satiety. These are some of the hypothalamic and some are metabolic. I talk about anorexia nervosa, bulimia nervosa, hypothalamic obesity, Pickwickian syndrome, and some of the obesities are hypothalamic. Fever and loss of appetite, again hypothalamic centers are involved. Cachexia, hypothalamic centers are involved. So these are the disorders associated with the feeding and satiety in relation to hypothalamus. Let us